because any of the donor chromosomal DNA sequences can be transferred to the recipient. All right, and then in chapter 13, we'll talk about specialized transduction. It's a little bit more detailed. It actually involves 2 different replication cycles in the viruses, but we aren't going to go there yet. However, this does permit us to describe or answer the question posed in your textbook about different strains of E. coli. So I actually want to take a little time out, you guys, to explore the evolution of this toxin-producing strain of E. coli. Because this toxin-producing strain of E. coli is a major problem here in California, and we want to be aware what is the reservoir, how would we get infected, which people are at really high risk for possible death from this toxin-producing strain of E. coli. Okay, now you might say, why are we talking about this? Well, we're going to see that it is through transduction that this particular strain of E. coli acquired its toxin genes. So the strain of E. coli that we're describing is E. coli serotype 0157H7. Okay, folks, so from chapter 9, motility in lab, help me out. What is this H antigen? What part of the bacterium is the H antigen? Good, excellent. This is the flagellar antigen. And remember, the flagella are made out of proteins. And proteins are wonderful antigens. An antigen is any substance our immune system sees as foreign, and thus will trigger a specific acquired immune response, such as antibody production. So we can see amongst the E. coli, there's at least 7 different proteins that different E. coli would make. 7 different flagellar proteins, and our bodies would make specific antibodies that could recognize, we make a different set of antibodies to recognize each of those 7 different flagellar proteins or antigens. Okay, and do you remember what the O antigen is? What part of the E. coli is the O antigen? Oh, excellent. That is so good, you guys. It's the polysaccharide of what? Oh, you are so good. Yes, a lipopolysaccharide. And where do we find lipopolysaccharide?
In the outer membrane.
Good. Yo, you're nailing it. The outer membrane of food. Gram-negative bacteria. You guys are doing dynamite. Okay. So it turns out polysaccharides they're not very good antigens. They don't trigger a really strong immune response, but you can get some antibodies made against them. So we can use these antibodies to identify specific strains of E. coli, right? So here we know that in E. coli, it can make 157 different types of O antigens, right? So again, we can use antibodies to identify specific strains of E. coli. And then this then becomes the serotype. Sero from serum that contains antibodies, using antibodies to identify specific types or strains of bacteria. Now, this is important because you know right now we're all chock full of beneficial E. coli in our intestines, right? They aren't hurting us, they aren't making toxins. But some E. coli have evolved or acquired very powerful toxin genes, and this is one of them. Now, the reason we're so worried about this guy has acquired a Shiga-like, break this down, a Shiga-like toxin gene. And this Shiga toxin is so powerful. Okay, so the E. coli, that E. coli 0157H7 has a Shiga toxin gene in its chromosome. And the Shiga toxin gene encodes this incredibly toxic protein toxin called the Shiga-like toxin. And this toxin, what it does is it inhibits ADS ribosomes. So what function would the Shiga toxin knock out in our cells? Protein synthesis, right? And protein synthesis is really important for our cells to maintain themselves, for repair, for growth. So if we were to swallow this E. coli and transmission, you guys, and we'll get into more of this, is fecal oral. These bacteria, they live in the intestinal tract. So they're shed in feces. So wherever the feces goes, the E. coli goes. If we were to swallow fecal contaminated food or drink fecal contaminated water with E. coli, it colonizes the intestine, the colon. And infections, very often the first clinical sign is a bloody diarrhea because of damage that's occurring to the intestinal mucosa in the blood vessels. So you can get frank blood. Frank blood meaning it's red, right? That's telling you you're getting bleeding in the lower intestinal tract. The blood hasn't been digested. It's frank blood. And this is really serious stage, especially in little children. Little children can die from this. So I would recommend, if you guys have kids or your friends have children or nieces and nephews, if they ever develop a bloody diarrhea, I would run to the hospital and I would insist that they include this toxin-producing strain of E. coli in the differential diagnosis. Okay, and the reason is that a small percentage of people that become colonized, the Shiga toxin gene is absorbed into the blood vessels where it can cause blood vessel damage. The blood vessel damage can eventually cause lysis of red blood cells or erythrocytes. The toxin can circulate to the kidneys and cause kidney damage. So it is a small percentage of people that get colonized, but this can be life-threatening. This collection of abnormalities is called HUS, and it stands for hemolytic uremic syndrome. And this is life-threatening. This can kill. This can especially kill little children. So hemolytic because of the lysis of the red blood cells. So you can end up with an anemia, right? There's all the red blood cells are getting lysed. Uremic means high levels of urea in the blood. That's an indicator your kidneys aren't functioning well. It's suggesting kidney damage. That syndrome, again, we have multiple things going on. This can be fatal. Now, why are we talking about this? Well, it turns out this is an emerging pathogen. Prior to probably the 1950s, prior to World War II, this strain wasn't making toxin. So the question is, how did it acquire this toxin gene? And through DNA sequencing, they think they know. So it turns out the original Shiga toxin gene came from a close, this would be the donor, recipient. So the donor bacterium of the Shiga toxin gene is a close cousin of E. coli. It belongs to that same big family, the Enterobacteriaceae. So Shigella dysentery is a close cousin of E. coli, and Shigella dysentery causes bacillary dysentery. And again, the original donor of the Shiga toxin gene was Shigella dysentery. So the question is, how did, say, prior to the 1940s, how did this non-toxin producing strain of E. coli 0157H7, how did it acquire the Shiga toxin gene? Well, through DNA sequencing, they know absolutely for sure it was through transduction. And what is transduction, you guys? How did the donor Shiga toxin gene get introduced into our originally non-toxin producing E. coli? Yes, by bacterial virus. And again, we'll try to come back to this. This is actually an example of that more complicated type of transduction that we'll discuss later, where the donor DNA in this more complicated type of transduction is actually connected to the donor DNA. So in the recipient, the phage DNA and the donor DNA both insert into the chromosome. And again, we'll it's called specialized transduction, more complicated, we'll come back to it. But the reason that's helpful is because the reason they know with great certainty this is how E. coli acquired the gene. When they do the DNA sequence of the toxin-producing E. coli 0157H7, they find the DNA sequence for the Shiga toxin gene, and right by it is a phage DNA sequence. So they know it was a phage that brought the toxin gene to the E. coli and actually helped to get it inserted into the recipient's chromosome. So again, you're like, well, who cares? Well, why are we talking about this? This process, you guys, it's continually happening. It's continually happening. And so just so that we're aware that in 10 years, you'll be dealing with brand new bacterial pathogens that we don't even know about today because they haven't evolved. So we're going to see new bacterial pathogens evolve through horizontal gene transfer, right? So you guys, you're never going to be bored. If you're going into any aspect of health sciences, medicine, microbiology, you will never be bored because these guys are always going to be optimistic. You know, they're always like, what can we do next? I'm going to do next. Okay, so now we know the answer to that. Oh, some follow-up for us just with regard to natural history. So here in California, this strain of E. coli is really important. So just a little bit of history. This strain of E. coli, a very common reservoir is a source, are cattle. And so the E. coli lives in the cattle intestine. And what's interesting is the cattle don't get sick. And it's believed the cattle cells lack the receptors for the toxin. Shigella dysentery evolved as a human pathogen, so that the toxin binds to receptors on human cells. It doesn't bind to receptors on cattle cells, so the cattle don't get sick, which is bad news for us, because we don't know they're carriers, right? So a couple of things can happen. Initially, back in the, let me see, was this the 60s or the 70s, appearance of E. coli 0157H7, we started seeing outbreaks associated with hamburger. It was often known as the hamburger disease. And the reason is, and this is disturbing, you guys, but it is good that we know where our food comes from. So when cattle are sent to slaughter, at the slaughter yards, they know something's really, really wrong. So they go into fight or flight, and they lose control of their bowels. So at a slaughter yard, there is literally feces flying everywhere, right? So after the cattle are killed, the folks that are doing the butchering, they are trying so hard not to contaminate the carcass. So after they take the hide off, the skin off, and you just have, you know, this carcass, lots and lots of meat there, they're trying really hard to make sure feces doesn't contaminate the carcass, but it's almost impossible, right? So the idea is after slaughter, this is your carcass. It doesn't have skin on anymore. But you guys just presume whenever you're handling raw meat or raw poultry, just presume that the outside, there's a fine covering of what? Yeah, fecal contamination. Just presume that. So if the feces has E. coli 0157H7, just presume it's going to be, you know, all over. Okay, so now we want to ask a couple questions. What happens if you, okay, and I am a meat eater, you guys. I've tried really hard to become vegetarian, but I'm just too lazy. I haven't tried hard enough. I still eat meat. I love meat, okay? But so let's say I was lucky and I got to eat a steak, a solid piece of meat, right? So here's my steak. Where would the fecal contamination be on a solid cut of meat? It's just going to be on the outside, right? So consequently, as long as I cook or sear the steak, get the outside hot enough, it will kill the E. coli. Now, luckily, you guys, the E. coli, they aren't invasive. So the E. coli don't have enzymes to digest their way into the middle of like a thick steak. They're going to remain on the outside. So consequently, as long as I sear the outside, I could eat that steak medium rare, right? I don't have to cook it all the way through. I just need to make sure that I heat the outside sufficiently to kill any of the fecal pathogens. And E. coli it's killed easily by heat, right? But the problem is, you know, if you are a meat eater, if you don't have a lot of money, what do we usually end up eating? Ground meat, right? Ground meat, where they take odds and ends. So ground beef, what we traditionally call hamburger. Okay, so here's my ground beef. I'll make a hamburger patty here. Now tell me, where is the fecal contamination? Just on the outside?
Everywhere.
Everywhere, right? When you grind meat, you're taking that fecal contamination and the fecal pathogen and you're grinding it up. So our E. coli are going to be everywhere, on the outside, on the inside. So what would happen, you guys, if this was a hamburger patty and I cooked it so it was only medium rare, meaning I don't cook it all the way through? Is that safe?
No.
No, that's not safe. Because medium rare, your E. coli, your fecal pathogens are still going to be alive inside. So the take-home message here, folks, is especially in little children, if you're going to give them hamburgers, as a parent, I would break it open. And if it's pink in the middle, don't let your kids have it, right? And a lot of times adults can handle it better, but little kids, these infections, they can be potentially lethal. Older folks, really old folks, they can be lethal. Anybody who's immunocompromised. So, and hamburgers are fast food, right? We're getting it because, we want it fast, and unfortunately sometimes the restaurants are cooking it too fast, right? So they don't cook it all the way through. The other thing you have to watch out for, you guys, it might be that the hamburger is being cooked properly, but then after it's cooked, plates, surface areas, utensils that have come into contact with the raw meat are used for the cooked meat. So you re-inoculate it with the pathogens, the fecal pathogens. So you guys might be too young, but did any of you remember the Jack in the Box outbreak? Yeah. So this was an outbreak of E. coli 15787 where several people got infected. And I think there's at least a couple of children died from it, right? So that was, this is just off the site. They got such bad press, obviously, right? They were using so much business. They had an advertising company come on board, and that's where Jack came from. Remember Jack that's now in all the commercials? He was their response to this disaster of the E. coli outbreak in the hammer. And I like Jack. He always makes me laugh, but I didn't realize he was a result of the E. coli 15787. Okay, a little bit of nerdy microbiology history. The other thing, folks, why this is important here in California is that over the years, we're starting to see more and more of these toxin-producing of E. coli in fresh fruit and leafy greens, things that we eat without cooking. So now we have to be really, really careful of salad greens, any kind of salad component that won't get cooked. So lettuce, sprouts, spinach is another big one, and also fruits. Because again, you guys, it's fecal contamination. So one of our latest outbreaks here in California involved spinach. And there was so much concern about how the spinach got contaminated. This was spinach that was put in a plastic bag, which are wonderful incubators for bacteria, pre-washed. Sometimes, what I'm about to say, just ignore it. But sometimes the wash water gets contaminated with feces, so your salad greens are getting a nice fecal wash. That's not true. The companies would sue me, but probably in the past that did happen. But the spinach was bagged and it advertised as pre-washed, so people were using it in their salads. They weren't cooking it, and it had the spray E. coli 157E7 in it. So there was a lot of investigation where the contamination come from. There were dairy farms, so it's possible if cattle manure gets into water and that water is used to irrigate the vegetable fields. What does irrigate mean? To water, right? So people contaminated irrigation water could be a source. What they finally decided was it was wild pigs. The spinach field was fenced off, but wild pigs are pretty aggressive. Wild pigs had broken through the fence. They went in and eating spinach. They know it's good for them, and they poop. So it's not just cattle that can carry it, wild animals can carry it. Maybe the pigs picked it up at the dairy, I don't know what, right? But they think it was the wild pigs that brought it in. They pooped, and then the spinach got contaminated. Can't do that.
About 7 years ago, I used to work for Biostatistics, which is part of the Department of Human Health Services, and there was an outbreak of E. coli in the lettuce.
In the lettuce.
Yeah, and they took samples from people's homes, the whole 9 yards, to trace where it came from. And it's so amazing how they found out it came from like Salinas,
Selena.
and it was
Salinas.
human E. coli.
It was human E. coli, right? Yeah, and that's another, thank you so much, Angela, for bringing that up, because not only can animals carry it, you guys, healthy adults may carry it and might not have dramatic signs, maybe a little bit of diarrhea. So anybody processing the food, the beef, any of the greens or fruits, humans can be the source of the fecal contamination.
It was the workers.
Yeah, right,
Yeah.
And that's really tough. So just like you're saying, it's so important that they find out what was the source, right? How can you prevent it if you don't know what the source was? So we have animals, we have humans, right? So feces gets everywhere, you guys. Yeah.
So when you just rinse, should you do it under a certain condition? Should it be like hot water versus?
Okay, now what I'm going to tell you is what the extension specialist over at UCD told me. He said, if you are feeding at-risk populations, little children, older folks, anybody who's immunocompromised, this is what he told me. He says, just cook it. And the reason is, and I don't mean to make you guys paranoid, like somebody said after taking this course, they don't want to go out, they don't want to eat anything, they just want to hide in their bed. But just a study, just so y'all know, just to make you truly paranoid, what they've done, the USDA did studies with like lettuce and spinach. And what they discovered, they immersed the leaves in solutions of E. coli 0157A7. So right off the bat, you guys, it's kind of extreme. But what they discovered is, this is goofy, too big, but leaves have little stomata, little openings for gas exchange. And what they did after immersing the leaves, they pulled them out and then they looked at it with the electron microscope. And what they discovered was the E. coli could actually get into the inside of the leaf through the stomata. So that means any amount of washing, you're not going to get them out. And another thing they said was that anytime there's a break in the leaf, like you go to the market and it's broken or torn, that's a wonderful entry point. Torn leaves, broken leaves are wonderful entry points for E. coli. So once they get on the inside, you can't get them out. Now, you guys, we need to remember, usually our leafy greens don't have fecal contamination, right? Industry is obviously very, very invested in trying to make sure there's no fecal contamination. So I still eat leafy greens, spinach, lettuce, all of that stuff, and I don't worry about this. I just rinse it under the tap, right? But I do give thought, if it's a high-risk person that I'm feeding, then I'm a little bit more careful. But for you all, you've got robust, healthy immune systems. Keep your ear peeled. If there's an outbreak of E. coli, right, you're going to be very careful. But normally, I wouldn't be neurotic about this. But just so you know, if there is contamination, even rinsing might not get rid of like E. coli that's on the inside of the lettuce leaf. But that's kind of overkill. Like I said, I've got California lettuce down in my lunch right now, big bowl of it, yeah. But again, high-risk groups. That's what I always think about, high risk. I'm gonna do it differently for somebody in a high-risk group. Good questions, you guys. Okay, so I know that was a lot, but big issue here in California. Okay, so we're gonna finish up, you guys, with a very brief discussion of plasmids and transposons. Oh, and now you got me started. Just like quick telling stories. Okay, but they've also done studies where they found bacteria like inside vegetables, and what they think is happening is as the seeds are coming up through dirt, it could have feces in it. So the plant itself could have like fecal pathogens on it, and they think in some cases where like the vegetable has a flower. Sometimes fecal pathogens, I don't know, from the dirt, from the bees, I don't know who's bringing them, can contaminate the flower, and when the flower is fertilized, it forms the vegetable around it. I think there was a study where they found enteric bacteria inside tomatoes, and it wasn't that they had invaded from outside, they think that the enteric bacteria were there when the fruit formed itself. And again, you guys, that, it makes us crazy, then we can't eat anything, but it's really fascinating as they try to unravel how our food becomes contaminated. It's pretty fascinating, all the different ways that it can occur. Okay, so now let's just finish up with plasmids. Can you guys give me a simple definition of a bacterial plasmid?
Carries genetic, extra genetic.
Good. Function, it's going to carry extra genetic information. Where is it located? It's usually, by definition, it's not part of the chromosome, right? So we call it extra chromosomal, meaning outside of the chromosome. And like Rochelle said, extra as in extra genetic information, usually genes that help a bacterium survive in a given environment. And plasmids, they have their own origin of replication. You're going to have multiple copies of a plasmid in a bacterium, and so they are wonderful, wonderful sources of donor genetic information. Bacteria love to share their plasmids with their neighbors. And a group of plasmids that we're really worried about are resistance, or R plasmids. Resistance, because these are plasmids that carry genes for antibiotic resistance and or resistance to heavy metals. And the heavy metals, you might say, what the heck is that all about? In the old days, before the discovery of antibiotics, heavy metals were a common antimicrobial. So, for example, mercury was often used to treat syphilis. Silver was used to treat bacterial infections. So there's been natural selection for bacteria to evolve genes that can help detoxify some of these heavy metals. Well, many of these R plasmids may carry multiple antibiotic-resistant genes or heavy metal resistance. So let's just take a peek here, you guys, and we'll come back here, I promise. So this is a great diagram of a plasmid. This is a map of a plasmid. And what's so disturbing, you guys, about this plasmid, twofold. Let's look at all of the resistant genes. So MER, this is for mercury resistance. SULF is for sulfa drug resistance, the inhibitors of folic acid synthesis. STR is streptomycin. This is an aminoglycoside that knocks out 70S ribosomes. This is streptomycin resistance. CML is chloramphenicol resistance. Again, chloramphenicol knocks out 70S ribosomes. And down here, tetracycline is another 70S ribosome inhibitor. So let's count all the resistance genes. So we've got 1, 2, 3, 4, 4 antibiotic, antibacterial resistance genes, and a heavy metal gene. So this single plasmid carries resistance to 5 antimicrobial compounds. Now, what makes this a nightmare? A nightmare plasmid for us? It's also a conjugation plasmid. What do you think a conjugation plasmid is? Have we talked about a conjugation plasmid? We have, but we just didn't call it that. Can you think of a plasmid that we talked about that permits a bacterium to conjugate? Good. Is the F plasmid a conjugation plasmid? Does it carry the genes that permits the plasmid owner to conjugate? Yes, it does. And here's a nightmare, you guys. This is a conjugative R plasmid. What does that mean? It carries the genes for conjugation and it carries genes for antibiotic resistance. So this means this plasmid carries the genes to permit its transfer to neighboring bacteria.